All right, so we're going to move on from fungi and fungal-like organisms into something a little bit more simplistic. So our bacteria are our prokaryotes, all right? And these organisms, while they do not have as many structures that they produce because they're single-celled organisms, they still have a lot that goes into them. So we're going to discuss the actual bacterial cell and what this means for how bacteria are going to be able to enter and exit hosts. All right, so we'll just start with, if my pen writes, we'll start with a simple bacilliform, pill-shaped bacteria, okay? And what I just drew, I'm going to say is our plasma membrane, okay? So the purpose of the plasma membrane is to allow transfer of different molecules in and out while still providing the basic structure so that all of the innards don't just come pouring out into the outside world. Okay, so every cell in your body also has this plasma membrane. What bacteria then have, and I know you are all going to appreciate this as you try and draw these yourselves at any given time in your career as undergraduates and graduate students. The next circle that I just drew on the outside of this, okay, that's going to be your cell wall. The cell wall is going to provide more rigidness. It's going to provide a little bit of protection, and it's going to keep it in a certain shape. Now, the density of this cell wall is what's going to make a bacteria gram-positive or gram-negative. But for purposes of this class, you don't need to know what that means. Just know that bacteria also can be characterized based on the way that their morphology looks. Okay, so gram-negative and gram-positive bacteria has to do with that cell wall. The next layer, which I'm going to switch up just so that it's not all the same color. Some people will call this the capsule. But this is known as the EPS, or the extracellular polysaccharide. So this extracellular polysaccharide is going to be gooey, all right? It's going to serve more as something that allows it to form a biofilm, where you have colonies of bacteria that are sticking together, mostly inside of some kind of vascular system or on the surface of a plant or on the surface of a table even, because remember, bacteria are not just plant pathogens. There are human pathogens that you would worry about during the winter months that still use these same tactics. The other thing that you may or may not know is that this extracellular polysaccharide, or this gooey, snotty substance that's on the outside of these bacteria, is used in a lot of your foods. So it's, in other words, it keeps all your foods stuck together. And if you see xanthan gum on anything, that's what you're eating. You're eating that extracellular polysaccharide that's sticking all your foods together. The other thing that bacteria have to help them attach and also to sense the world around them is pili. So these are short little hair-like things, right? Appendages, I guess, if you want to call them anything. I'm drawing way too many in order to make this succinct here, right? But these will help them stick together, and it'll help them when they are trying to, I guess, sense the world around them, whether or not they have bumped into something or not. Gonna switch up. The other outside of the cell organ, I guess we'll call it, for lack of a better word right now, is going to be the flagella. Now this one has multiple flagella. Sometimes they only have one. This one's also polar flagella because it's on one end. It can also be paratrichous, which could be everywhere. It could be bipolar on both the sides. Okay, again, these are all characteristics that distinguish bacteria from each other. On the inside of the bacterial cell, you have the chromosome, which is made of DNA. This is your genetic material. This is what's going to allow the cell to produce everything else it needs to survive, just like in your cells. Okay, except that it typically only has one chromosome. And if it needs extra, it has things called plasmids. Plasmids are little circular pieces of DNA. Often these are what contain the antibiotic resistances. And bacteria are able to trade plasmids back and forth with each other. While they're still all asexual, single cellular organisms, they can then say, oh, I have this very useful plasmid, and I think my neighbor might like this very useful plasmid that protects me from this antibiotic. And they can give their neighbor that very useful plasmid. Now, the one benefit that we have as plant pathologists is that they have a limited capacity on how many of these plasmids they can hold. So if for some reason they have an antibiotic resistance and they need to don't come into contact with that antibiotic for a long period of time, and they need a different plasmid, they'll discard the one that they think is no longer relevant in order to have a new plasmid that is more relevant for them. And then those antibiotics would start working again. All right, so this is why it's really important that you rotate any antibiotics that you'd be using. It's also why it's very important, as if you're thinking of human pathology, that you just don't continually shove the same antibiotics down through the population, so that these bacteria eventually will lose those antibiotic resistances. Okay. Inside here, this whole matrix is called the cytoplasm. Okay, I know that sounds really basic, but the cytoplasm is actually very, very important, because the cytoplasm is going to keep everything inside at the correct pH, all right, so that everything functions properly. This is why we just don't have our cells filled with water, right? Because we actually don't want our cells to be at the same pH as water at what, 6 to 8, right? There's actually a very specific pH that each type of cell needs inside. So the cytoplasm will provide that. And then the other thing that they have inside that often we forget about is that they have ribosomes, because they need to be able to produce protein. And that's what those will be for, if they need to produce protein. We often forget about them because we say that bacteria don't have organelles, and that's not true. They don't have membrane-bound organelles, right? So there's no nucleus in here, there's just a chromosome, and you won't see mitochondria or anything in here, but you will see ribosomes, because those are not membrane-bound organelles. All right, so now that we've discussed what bacteria look like and what they have at their disposal, it's obvious that they have limitations that, say, fungi don't have. So these bacteria are not going to be able to directly penetrate anything, okay? They're going to have to get into plants indirectly. Now, this could happen through the roots, it could happen on the leaves, all right? There are plenty of indirect spaces, so why don't we talk about that for a second? So we're just going to use a leaf for a second. And I know, my lovely leaf. Okay. And on the surface of this leaf, we're going to have some bacteria. Because believe it or not, bacteria are often epiphytic. Okay, so they're on top of plant surfaces. And for the most part, they're not pathogens by nature. So bacteria are what's known as facultative parasites, which will mean when they're put in a situation, they will transfer their mechanisms so that they can survive as pathogens, but they're more than happy living saprophytically on top of the leaf. Okay? So some of the structures that are on top of this leaf that are going to allow them in, the most obvious one is going to be the stoma. Okay? So the stoma are where the plants are going to produce or do their air transfer. And they're little holes that open up on the surface. And as they open up, the bacteria might fall into the plant, at which point they're now going to have to survive as a parasite versus a saprophyte. Another thing that happens is during the night, plants take up a lot of water and they don't have the sun beating down on them as much in order to transpire. So they build up turgor pressure. And in order to prevent their cell walls from exploding, this turgor pressure has to be released. So in order to release the turgor pressure, there are hydrothodes on the outside of the leaf. And this is why if you were to go out in the early morning, you would see water on the outside edges of the leaf where these pores are, these hydrothodes. Okay? But as you have water pooling up on the outside of the leaf, the bacteria can fall into these hydrothodes. They won't be able to get back out per se, and then as the day progresses, that water gets sucked back into the plant along with the bacteria. The other thing that may occur is wounds. So let's look at our we have a little insect here, right? I don't know, a little blue insect, okay? And he goes and he'll create a wound as he's eating at the plant. And as he's eating at the plant, again, the bacteria will be able to get into where that wound surface is. None of this is direct. All of this is indirect, okay? So that, again, bacteria do not have the ability to directly get into into the plant. They can also get in the roots. Roots get wounded all the time. In fact, just to grow, roots wound themselves as they're growing. And there are some bacteria that can chemotax and find these different areas to purposefully get into the plant. Whether it be the stomata or a wound or the root exudates, they can get to that and get in on purpose. All right, I think that is all I wanted to say about bacteria. Think. Yes. Oh, no, it's not. Oh, pause. Once they get into the plant, okay? Once these bacteria get in, a lot of times what you'll see is that they'll form these biofilms inside of the vascular tissue. All right, so bacteria are not often just going to spread from cell to cell to cell to cell because, again, they can't break through that cell wall. But if they can get into the vascular system, they can then clog that up, which is why you'll get things like wilt and scorch associated with bacterial diseases as the bacteria huddle together using the extracellular polysaccharide and those pili in order to have these large masses inside of your xylems and phloems. And that's actually everything I want to say about bacteria.